Okay. So there's people in the waiting room. Could be.
Yeah, we'll let everybody in around
Okay,
the hour.
okay. So yes, mainly what I'm going to be showing is the data that is already published in a paper that we published last year. So there's no problem.
So Natalie, I put the links to those things in the document as well. That's the open document for this group. I can drop that in the chat. You can take a quick look at that if you want. But then that will also mean we can just point people to the open document.
That's perfect. Yeah. I think we've let everybody in from the waiting room so they can see the chat and open up the Google Doc. But as more people come in, I think we'll need to resend that again.
I can keep posting that in there.
Thank you.
And if everybody would like to add their names, there's a spot for that if you want.
So we can give it a minute or two, and then I'll get started with the introductions. All right, so I think we can get started. So hi, everybody. Thank you all for being here today. And I guess Happy New Year. It's been a while since we've last met. A few things we've put into the chat, our ongoing notes where we would like everyone to put in their names just to have an attendance. So if you could put your name and affiliation, that would be great. And so Nikki has just added the link to that Google document into the chat. And this is an open document that we continue to populate, so anyone can put in any notes or any thoughts and comments throughout the presentation. But we will also have somebody taking notes for those who cannot make it today. We just had a few more people join in, just in case you missed it. We're all just adding our attendance list. So if you can put your name and affiliation into the Google Docs linked into the chat, that would be great. And this meeting will also be recorded and uploaded for future reference, so you'd be able to find the link in the Google Docs. So just a few other, I guess, announcements before we begin. First, we have a resource. There's an open microscopy environment community meeting, and early career BINA registrants can apply for BINA support to attend this, and this will be closing on February 18th. So if you're part of BINA and you would like support for this program, please apply. And there will also be a GBI BINA annual meeting on imaging in 2035, sustaining infrastructure ecosystems and advanced technologies. This will be happening in Montreal in October of next year, or well, this year. And so please click onto the links into the ongoing chat to learn more about what's happening. And we'll also be having an expansion specific session for this meeting. So it would be very relevant and interesting. And yes, so thank you, Nikki, for putting that in the chat. Applications for travel and registration support are also already or will be open March 5th or until March 5th. Perfect. So without further ado, I would love to introduce our guest speaker today, Dr. Ramiro Tomasina. He'll be talking to us about his talk, From Trypomastigotes to Trypomastigotes, Analyzing the One-Way Intracellular Journey of Trypanosoma cruzi by Ultrastructure Expansion Microscopy. And this was work that has very recently been published. Dr. Ramiro Tomasina is a postdoctoral researcher at the Institute Pasteur de Montevideo, Uruguay, and an assistant professor in the Department of Parasitology and Mycology at the Faculty of Medicine, University of the Republic. He is also a member of Uruguay's National Researcher System, and his research focuses on host-pathogen interactions with a particular emphasis on Chagas disease and cell division and Toxoplasma gondii. So he completed his PhD in 2023 at the University of the Republic, where he studied centrosomal proteins and Toxoplasma gondii. Following this PhD, he joined the Host-Pathogen Interaction Laboratory at the Institute Pasteur de Montevideo, which was led by Dr. Carlos Robello. The Robello Lab has a diverse research group dedicated to studying the parasites, Trypanosoma cruzi, Leishmania, Toxoplasma gondii, and Neospora caninum, as well as the host response to infection. So since joining the lab, Dr. Tomasina has worked on topics ranging from fundamental biology studies to involving animal models. So thank you so much, Dr. Tomasina, for being here today, and I'll pass the floor on to you.
Okay, thank you, Natalie, for the nice introduction. Hello, everyone. Thank you for this opportunity. I hope at the end of the talk you understand why, at least the title and what is this about. So because it's easier for me to pronunciate, I'm going to just instead of saying ultrastructure expansion microscopy, I'm going to say just UXM. It's easier for me. First of all, I have to introduce the parasite. Who is Trypanosoma cruzi? And Trypanosoma cruzi is a protozoan parasite, so it's a unicellular parasite, eukaryotic unicellular parasite that is the causative agent of Chagas disease. It affects millions of people in Latin America, and it was first described by Carlos Chagas, which is who was a medical doctor in Brazil, and he described it at the very beginning of the 20th century. This Chagas disease has been historically restricted to the Americas, but I say historically because nowadays we would say it's a international concern. It's a disease that is turned to worldwide concern. So why we studied Trypanosoma cruzi? Because it's the causative agent of Chagas. And I took this flyer from this website down here because I really like the title. Chagas is a silent killer because this disease, it has 3 clinical phases, which is the acute phase where there are few symptoms or even can occur without symptoms, where the parasites are getting inside the organism and they are multiplying and they are growing in number. Then the immune system response occurs, and we got the patient go inside a chronic asymptomatic phase where there are no clinical symptoms, and then after 30 years, the patient can develop chronic clinical phase, which involves a part of the patient infected, and they develop heart and digestive system problems. So it's a silent killer because during many years the patient can even notice that he's infected with this parasite. An important thing is that this disease is transmitted by a vector, and so well, the vector has been restricted and controlled in many areas of America, but is still a problem, especially in poor areas. So if we go to the life cycle of Trypanosoma cruzi, we can observe that to fulfill this life cycle of this parasite has to go on to two kind of hosts: the vector one, the invertebrate host, and a vertebrate host or mammalian host. So this parasite is going to face 2 different environments, and so it has a lot of challenge to overcome. In the vector, if we focus in the vector, we are going to see that a vector can get infected from a mammalian host that is infected with the parasite by taking a blood meal of an infected mammalian host. So when it takes the blood, it gets infected with a life form that is called Trypomastigote, and this Trypomastigote goes inside the digestive system of this vector and switch to another life form, which is Epimastigote. These epimastigotes are replicate forms, so they multiply and they grow in number in the vector, and then they switch at the very last part of the gut of the vector to a metacyclic trypomastigote. This is an infective form and not replicative form. This one is the one that is going to be present in the feces of the vector, and it's the one that's going to infect the mammalian host. How is going to happen this? When the vector is taking a blood meal, it's going to make poop. I don't know if it's clear. So in the feces of the vector, we're going to find the metacyclic trypomastigotes, and that one is going to infect through the wound that the vector has already done, is going to infect the mammalian host. And this mammalian host is going to get infected with the metacyclic trypomastigote, which are the infective stage of the parasite. And this metacyclic trypomastigote are going to start infecting the cells of the tissues that are going to reach. And once they infect the cell, they are going to switch to a replicative form, which is a mastigote, but this time intracellular form, and they are going to multiply and they are going to grow in number, and then they're going to start differentiating to trypomastigotes that are going to be released once the cell is sliced out. So once the cells is sliced out, we have the trypomastigote that are going to be released by the cell, and these trypomastigotes are going to infect other cells and so on. So when this cycle will finish, or at least slow down, when the immune response is active. So once the immune response is active, this cycle is going to slow down, and we're going to find the mastigotes that are going to stay there, but dividing slower, and this cycle is going to be slower in heart tissue and in some brain tissue as well, and in the digestive system tissue as well. So basically, that's what is going to happen with the cycle. So of course, the cycle is going to be finished once the trypomastigotes are going to be swimming in the blood, and the vector is going to get infected once another vector gets infected once takes the blood meal of that infected animal. One thing to keep into consideration is that the parasite, depending, no matter the host, whether it's the invertebrate or it's the vertebrate host, does the same strategy, which is goes to a life form that is a replicative form. So in here we have the epimastigote, and in here we have the amastigote. So they grow in number and they colonize the vector, the host, sorry. And one thing to highlight from here is that in this switch from trypomastigote to amastigote, we have important differences in the shape and the organization of the cell, of the Trypanosoma cruzi cell. So we come from a very elongated and long flagellated form, which has the kinetoplast and the nucleus, and the kinetoplast is posterior, in the posterior part towards the nucleus. And then we have the amastigote, where we have a rounded shaped form and a short flagella, and the kinetoplast is in the anterior part towards the nucleus. So that is important to keep in mind, just to understand the following things. So basically, we have two kinds of life forms in the mammalian host, and then we have two kind of life forms in the vector, the epimastigote and the metacyclic trypomastigote. And in here we have the trypomastigote and the mastigote. But real life is always a little more complex. And this guy, who is Carlos Chagas, the one that described the disease and described the parasite for the first time, he figured out that by just regular light microscopy, that there were several shapes that this parasite was producing. And this is just part of the original paper. This is a figure from the original paper. And he observed all these life forms in the vector. So with my boss, Carlos Robello, we said, why don't we go deeper with UXM to try to understand how in this part of the life cycle, the changes are happening. So I had some experience with the UXM before, during my thesis. This is the first paper published, and where they described this protocol in 2019. And I remember the first time I saw this, it blew my mind, because I was working with some structures that were at the limit of the confocal resolution. So I said, well, maybe someday. And then this paper from Nicolás dos Santos and Dominique Soldati group came with the application of the UXM for Toxoplasma gondii, and that was the parasite I was working on. So we were able to work with this UXM protocol, and then more papers came. There was a work from Plasmodium as well, from Sabrina Absalon. There were papers in Cruzi, Victoria Alonso and Esteban Serra group. In the Esteban Serra group, they put the UXM apply for Trypanosoma cruzi. So we said with my boss, why don't we go and explore these changes by UXM, and as well these changes. So the first thing we wanted to observe was we wanted to have a closer look of how UXM could help us to observe really quick events in the life cycle of this parasite. So focusing on the mammalian host part, we said, why don't we check if UXM can allow us to observe the invasion process, which is super fast, because in half an hour we put the parasite in a monolayer of cells, and they, in half an hour, the majority of them are inside the cell. So we said, maybe we can check this by UXM. Maybe we can check how the parasite get inside, and once get inside, what it does and how it looks. And then we said, why don't we check these changes between trypomastigote and a mastigote, which is super fast as well. And it was a process that was poorly documented. Like, there was a gap in the knowledge of Trypanosoma cruzi life cycle at this stage, where the trypomastigote differentiates to a mastigote and how this was being performed. So we said, well, let's check if UXM can help on this. And then in this as well, because the parasite needs to get inside, differentiate to a mastigote, replicate, and then differentiate back to trypomastigote. That's why the one-way shortening of trypomastigote. So we said, why don't we get UXM in this window of time? This was more record because it takes longer. So there was more evidence with microscopy techniques. But in this window, especially of time, it was really, really hard to find something on the bibliography before this work to show how these changes were happening. So the first thing we did was to set up the experiment for the expansion. We checked the expansion factor was coherent, that we were able to expand the trypomastigote, we were able to expand the mastigote, and we try as well with other life form, which is the vector form epimastigote, just in case, just to see if the protocol was working on well. And that took us like around a month. And then we said, well, let's start the experiments. So the first thing we did was try to observe the invasion process. So this image is a 3D reconstruction using Agave 3D software. It's a free software. It's amazing software. And so basically what I did was we took set slides of an expanded sample, and in green or sky blue, we can observe the NHS ester, which is a reagent that reacts against all the proteins. So we have all the proteins that are in the sample, and with that we use it to show the shape of the cell. And then in gray we have the acetylated tubulin. So if we do a zoom in this window, we can observe that in gray we can observe this is the flagella of the parasite of a trypomastigote, and this is the posterior part. So basically what we are observing here is that the parasite is getting inside the cell, right? And as well, we can observe in here a parasite that is completely inside the cell. But so we were super happy to be able to observe this invasion process, because there are really a few pictures of this, and the only pictures that are available in the bibliography were from CM, scanning electron microscopy. So like this one, which was a paper from Norma Andrews. But with expansion microscopy combined with confocal microscopy, we were able to observe it, this invasion process. So we were super happy. So the next question was whether we were able to observe the trypomastigote inside the cells. And yes, what we are observing here is NHS ester in, again, this ester in red in green the acetylated tubulin of the parasite, and in blue the DAPI. So we again were able to observe what we proposed to observe, which was the Trypomastigotes inside the cell. This was quite exciting because there are a few pictures of TM of Trypomastigotes inside the cell. So we were able to observe the Trypomastigote just inside the cell after the invasion process. So how we know it's just the Trypomastigote just invade the cells? Because of the long flagella, the position of the basal bodies, we can even see the pair of the basal bodies, the kinetoplast, and the shape of the posterior part. As well, we can observe the elongated nucleus. So in here, we observe the kinetoplast posterior, in the posterior part towards the nucleus with an elongated nucleus. So we started looking at the sample, and what we found out was that after observing a lot of cells in the sample, is that apart from the Trypomastigote, the really long Trypomastigote shapes, we started observing these kind of life forms, which were a kind of Trypomastigote, but that was somehow getting closer the posterior part towards the anterior part, right? And it still have the elongated shape of the nucleus, and the kinetoplast is in the posterior part as well. And it has still, and it still has a long flagella. But it was somehow getting closer, the posterior part to the anterior part. So then we started observing these kind of shapes where, focus on this one, on this life form, where the posterior part was of the parasite was somehow touching the anterior part of the parasite. It was like all it was like all fold up. So like the parasite was like getting closer the posterior to the anterior part. So we observed many of these intermediate shapes, and with that, what we decided was to create again a 3D model using Agave, where we can Agave 3D software, where we can observe that in here, in gray, we can observe the tubulin, acetylated tubulin of the parasite. In sky blue, the DAPI, and this is the same parasite and the same 3D model, but we are showing different from different perspective. So what we observed was that the long flagella is getting surrounding the parasite, and then the posterior part is getting closer to the anterior part. As well in here, we can observe it. So we said, well, we are starting from a very long shape as the Trypomastigote is, and with a posterior, with the kinetoplast in the posterior part, and then we end up with the mastigote, which were the rounded shape, and with the kinetoplast in the anterior part towards the nucleus. So maybe we are observing the mechanism of the parasite, of how the parasite is relocating the basal body from the posterior part to the anterior part. So that was quite cool. And then we ask, what's happened with the corset of microtubules? Because this parasite, the shape of the parasite is because of the presence of a corset of microtubules that they gave the shape. So what happened with that? And what happened with the membrane? Because we have membrane in here and we have membrane in here. So what's going on there? Because there must be some membrane fusion, and what happened with the microtubules? So we did some, this was a quite hard moment because we couldn't find the answer for this, and we still don't have the answer, but we have an hypothesis. So checking the bibliography, really old bibliography in the 80s, this scientist, Norma Andrews, she found out that in many life forms of Trypanosoma cruzi, she observed in some places next to the flagella pocket, which is a part of the parasite that is close to the kinetoplast, she observed vesicles. And she said that maybe there is a fusion of membranes between the elongated shapes and the rounded shape. So makes sense with our hypothesis. And then what happened with the microtubules of corset and of the parasite? So checking another beautiful paper from the 70s, in this time, they did TM, transmission electron microscopy, of the Trypomastigotes, and they found out the arrangement of microtubules of each section of this Trypomastigote. And what they found out was that the amastigotes, they have 120 microtubules like this, around 120 microtubules. And then they did section by section of the Trypomastigote. And what they found out was that in the posterior part, where the kinetoplast is, P2, and the A1, which is the anterior part, they have 70 in the anterior part and 55 microtubules per section. So what we believe is that as this P2 part of the parasite is getting closer to the A1, well, we don't know for sure. This is just an hypothesis. What's going on is that there is a membrane fusion and then an intercalation of microtubules. So this, if we do the math in here, we are going to have 120 microtubules, so, which is the same as it has in the mastigote. So maybe we are wrong, but at least we have a theory of how this parasite changed from the elongated form to a rounded form, and with that, the parasite is able to assemble the mastigote, which is going to be the replicative form. So another thing that we observed in this work was that after observing this like intermediate shapes where it's a Trypomastigote that is getting rounded, getting closer to the posterior and anterior part, we observe as well this intermediate shape that has never been reported before, which is a rounded shape with a basal body position anterior to the nucleus, but they still have a long flagella. This was quite cool because we would say this is an intermediate life form between the Trypomastigote and the amastigote. And as well, if we observe carefully, we can observe that the nucleus of this intermediate form is elongated. So it looks like a nucleus of a Trypomastigote instead of the nucleus of a mastigote. So for sure, this was just really, this parasite was just, has already rounded, but it was super fast by the time we kept it and we could observe it. So after that, what we observe is just plain amastigote, which are rounded shape forms with a kinetoplast anterior to the nucleus and the nucleus that is really rounded. So just a summary of what I was telling you before, we were able to observe in detail the intracellular mastigogenesis, so the process of a mastigote formation from the time the parasite invades the cell and the parasite creates this intermediate form and then the mastigotes. And we were able to propose a model of how the basal body repositioning occurs. So after that, we went to observe in more, in longer infection times, because this takes time. The data I was showing you before, it's just a half an hour window between half an hour and 4 hours. After the 4 hours, between the 4 hours and the 24 and even 48 hours, we are going to observe amastigotes, which are the replicative forms. So basically, we were able to observe these round shape forms that were replicative. So as in here, we can observe 2 pairs of basal bodies and 2 kinetoplasts, and the nucleus is up to get divided. But one thing that really caught our attention was that we were able to observe 3 different kinds of mastigotes. We were able to observe round shape with a really almost not noticeable flagella. And then we were able to observe a round shape form, but with a really short flagella. We measured the flagella length, and we were able to observe that there was a statistical difference. And then we were able to observe another kind of shape, which is a leaf shape, like less rounded and with a longer flagella. And the 3 of them, they were replicative formed, and they had the position of the kinetoplast anterior to the nucleus. So makes sense that this was what historically were called as a mastigote. So what we did was to call them a mastigote 1, a mastigote 2, and a mastigote 3. But what happened after? After 48 hours, and between 48 hours and 72 hours, we were able to observe a diverse amount of shapes of Trypanosoma cruzi. So in here, what we are observing is in gray NHS ester as well, and we have in green the acetylated tubulin of the parasite. So if we do a zoom in of this, what we can observe is what resembles to the mastigote tree shape, but the cell body is bigger and it has a longer flagella. So we observe this intermediate form that has never been reported, and I will show you just how what we guess this life form looks at confocal microscopy. So the resolution increases amazingly. So if we go after 72 hours post-infection, this is a cell again in gray, and we have in green the acetylated tubulin of the parasite. What we can observe is that things were more complex, and it looks like a full carnival in here. And if we do a zoom on every parasite, we can observe different life forms. And in here, we were able to observe the epimastigote-like. They are called epimastigote-like because they look like epimastigote, the life form that is present in the vector, but they are a lot shorter, and I think it's 2.5 shorter than the vector one. And these were reported before, and we were able to observe the same and the same size. So we were quite happy with that as well. And then if we go further, we were able to observe trypomastigotes inside the cells that are up to be released by the cell that is getting destroyed by this parasite. Even if we observe in here, we can observe that these parasites are outside the cells, that are moving, just leaving the cells, and we were able to fix them just before. So how we were able to observe that and said this is a trypomastigote, this is an epimastigote, pretty much because of the size and pretty much because of the position of the basal bodies. As you can see in here, you have a posterior. I'm not showing the DAPI channel because it's quite confusing, but believe me, this is a posterior position of the kinetoplast towards the nucleus, and in here we have still an anterior position towards the nucleus if we see the basal bodies. So as a summary of what I was telling you, we were able to observe from the trypomastigote to a mastigote, and then we were able to observe the 3 different life forms of the amastigotes, or 3 different versions of the amastigote, and then how this amastigote differentiates to trypomastigote inside the cell. And we were able to identify this new life form, never reported at least until the paper. And we were able to check as well life forms that they were first described, like the epimastigote like with the shape and the size, which was quite as a internal control as well of what we were observing. And we were able as well to observe this life form, which is an intermediate life form, because it has the position of the basal body in between anterior and posterior part, and it's a life form that is in between the epimastigote, intracellular or epimastigote-like, and the trypomastigote form. So these are pictures that are present in the paper. As a summary, we were able to observe all these kind of different life form, which was a kind of our idea was a kind of trying to make a kind of atlas of what was going on with Trypanosoma cruzi inside the host cell. So we were able to have at least an idea of all the shapes that and structures we were observing. And it was, as I was telling you before, it was published in at the end of the year, or last year. So everything what I was talking is there. And well, that's the end of the talk. I want to thank the university where I work, the Institute Pasteur where I work as well, our laboratory, and the Department of Parasitology and Mycology where I work as well, and PSU and SNA because I belong to both systems, and they give us some fundings. So, and thank you all for listening to this talk.
Well, thank you so much, Ramiro. Those are some really, really amazing images. And it's crazy how you can just see it in that kind of resolution with a confocal. Like you said, it's literally comparable to SEM, which is the sample prep is so different.
Yeah, it's amazing. For us, it was a game changer, for sure.
Yeah, and seeing it in the color that it is, it's just so much more vibrant.
Yeah. Yeah.
All right, so we can open up the floor to any questions. Feel free to put any questions in the chat. I'd be happy to read them out loud or unmute yourself and we can get started.
I was going to ask if you could just say a little bit more about the sample prep and any challenges you might have faced in optimizing that.
Okay, thank you. The sample preparation has some difficulties, especially because if we want to get rid of the microtubules of the host cell, what we do is we follow the USM protocol that these guys from Soldati group were doing, which was basically we don't fix before, we just do the UXM, and like this protocol just gets away with the microtubule of the host. So with that, we get cleaner images. Of course, you lose some data, yes, but at least you are able to observe the structure of the parasite, which was our main goal. Of course, if you want to go for some host response or something, you have to optimize the protocol to be able to see those structures. But yes, I would say that's the main thing. And then basically I just follow the Toxoplasma gondii protocol, which is the one that is published, and I think it's the one that they use for malaria as well. And I did this. No, maybe not. And this one is Sabrina is saying no, and it's the one that I did for Brucia as well, and it works perfectly. But I don't know.
So how long does it take for you to prepare a sample and go to the
Well,
microscope?
it takes like 3 days. So you have to plan your experiment, and if the sample is not originally properly prepared, you spend like 1 week and you find out that it wasn't worth it at the end of the week. But well, it's worth it. You know, like it gives so much information that it's worth the time.
So there was a question in the chat from Alan Marone. Can you stain for the membrane of the parasite cell specifically?
Well, it's a good question. We don't have, I think we don't have any kind of antibodies against that, but it's something we can think, like we can try. For instance, the UXM is quite bad for membranes, so we know that for sure that is not the best protocol to follow. But there are some modifications of the protocols that if you fix with something before glutaraldehyde before, maybe you get better results. But I never tried. For this, I never tried.
And then another question from the chat box from Yurizada Rodriguez. Have you named this new parasite form?
No, it's just a plain, you know, a mastigote 1, 2, and 3, and that's it. And the intermediate form 1 and 2.
And another question from Andy. I may have missed it, but what's the best resolution you can get out of expansion in X, Y, and Z axons in your experiments?
Yeah, I didn't mention it, but UXM can reach between 3.5 to 4 times resolution. In my hands, I always check the expansion factor by measuring the shells and by measuring the size of the structure that we know for sure that they have to be more or less that size, and that is our control. But I don't know.
And then, so there was
Okay.
another question in the chat. Which of the helminths can be observed under advanced microscopy?
Sorry, I didn't understand
This
it.
was earlier. I missed it earlier, but in the chat it says, which of the helminths can be observed under advanced microscopy?
I have no clues. I don't know. I guess it's harder because it's a multicellular organism, and this is just one cell, so it's easier. Well, Sabrina has the answer as well, so she's more expert than me for sure. I don't scrub the host cell from the plate. I just prepare it in cover slips, 13 millimeters cover slips, and I just use the cover slip with the host cell. So I just detach it from the top of the plate, but it's just we have it in there, so I don't scrub the cells. I think if you scrap it, you're going to destroy the sample.
Hello, Ramiro. Thanks for this nice talk. I was wondering if you have tried to look at this difference in morphogenesis by transforming metacyclic trypomastigotes into a mastigote in axenic culture.
No, I never tried because that's something we have the full life cycle system. Like we invade get older the epimastigotes, so we get the trypomastigotes, and then we invade the host cells.
Yeah.
We
Okay.
never
Yeah. I
tried-
was wondering if it was really
No.
some feature were dependent really and influenced by the host cell, or it just the parasite itself that can do it also axonically.
Yeah, there is a paper from Normandius that they do that, and they observe in CM, so
Yeah.
maybe, and they observe something similar stuff, like they get rounded, and that's where I took the idea of the vesicles, that there is a fusion of membranes. She observed the vesicles in the flagella pocket. So
Thanks.
thank you.
Yeah, there were a few other questions in the chat. One is, from your experience, how long can the jellification solution last before it goes off, even if it's kept at minus 20 degrees?
Well, I know people, Sabrina has already replied. I know people that they kept their shells. I never put it into to work in our lab, I always do it and I just go an imaging. But yes, you can keep it like there are protocols for that. And there was a question because of how the epimastigote, if there is division. Well, according to our observation, we didn't observe any dividing epimastigotes. We think that the division ends on the final life forms that are a mastigote like leaf shapes. But that was according to our data. I couldn't find any epimastigote like dividing. But well, we don't know. For sure, like we counted and we couldn't find it.
Looks like there are a couple more questions in the chat. Sabrina has made a comment that says they have gels as old as 3 years and they are still perfect when they thaw them out. Thanks for sharing that, Sabrina. Oh, there's a question for you in the chat, Sabrina, about doing that. This has been great. Thank you. Really, really great questions and discussion.
Perfect. Thank you so much for being responsive on both ends.
Would you
Thank
be able
you.
to answer that, Sabrina, for the group about freezing and storing?
I answer in the chat.
Okay. Oh, you have a protocol. Okay. Excellent. We'll put that in the notes.
So thank you. Thank you very much for this opportunity to show a little about our work.
Thank you so much for coming and showing all these pretty pictures. It's great.
Great to see that this work is being done too, because it's an important thing to be tackling.
Maybe mention our next meeting too, Natalie.
Yep. So for the Canadian hosted, our next meeting will be April 2nd. We'll send out a meeting reminder a week before the meeting, as well as 24 hours before that, depending on where you are, so you can add it to your calendars. We will have Dr. Zenib Tenver from Rutgers University talking to us about the applications of expansion microscopy in neuroscience. So we'll be covering a very different topic, but still using expansion for a variety of applications. And yep, I think that's all that I had on my end.
Thanks, everybody.
Thank you.